So I do realize that what I'm going to talk about is not core population genetics, but that is for many years that I have done anything on population genetics, and so moved on to more organismal. Basically, my excuse is that I have done mathematical population genetics, and now I'm too stupid for mathematics, so I'm doing biology instead. And today I want to talk about the evolution of the fetal-maternal interface. And to do that, I want to just explain a little bit what the fetal-maternal interface is, in case this is not something that you are familiar with. So what we call the fetal-maternal interface is the connection between the placenta, which we conceptualize as the part that comes from the fetus, and the maternal side, which in mammals is mostly the decidua or the endometrium on the other side. And in many cases, the fetal-maternal interface is just an apposition of fetal cells and maternal cells, but in more developed and invasive placentations, like in the human, there is actually a interdigitation between fetal cells and maternal cells, as you can see here. So this is actually really a new tissue unit that is arising in evolution. So that doesn't seem to work. It's probably a little weak. Okay, so just to situate us here, we have mammals have three major groups: the monotremes, or the egg-laying mammals. Then there are the marsupials, kangaroos, wallabies, and so on, but also opossums. And then you have the placental animals that you better call eutheria, because otherwise you get into trouble with marsupial researchers who insist that marsupials also have a placenta, and therefore we cannot call them placentalia. So anyway, there's still so based on this simple phylogeny, you can infer that viviparity, or giving birth to live offspring, evolved in the stem lineage of Theria, and also a placenta evolved. But there are major differences that I will talk about. Namely, marsupial neonates look something like this, about the size of a small jelly bean. Many of them don't even have hind limbs at the time when they are born, and develop and grow in the marsupium of the mother attached to the teat. On the other hand, eutherian neonates can be quite developed, like in the case of an elephant, or with many hoofed animals. Now, it's interesting to think about what's actually cell biologically happening when a placenta evolves. So you start from a situation in egg-laying forms, also forms that have egg retention. You have maternal cells that have their evolved way of interacting with each other, and separated from them physically is are the embryonic cells that also have their own cell biology. Now, as an placenta evolves, the eggshell falls away, and all of a sudden you have cells that come into contact with each other and start interacting, which haven't seen each other for millions of years or ever in the history of life. And then all of a sudden new communication pathways open up. So for instance, if we have a maternal ligand that is produced to communicate with this cell, if this cell also has the receptor, they all of a sudden get a new signal from a cell type they haven't heard about before. So essentially, with the loss, or with the establishment of a direct physical contact between the mother and the fetus, sort of all hell breaks loose. Lots of new situations are coming about, and this situation is one that is a hotspot for the evolution of new cell types, for new ways of communicating with each other, as we have heard this afternoon by Dr. Cardoso this afternoon. In UBB, this is a place where new cell types arise at a high rate, and therefore it's a really interesting evolutionary situation. to analyze. Now, having realized that this is an interesting problem, we collaborated with Michaela Pavlichev's lab and my lab at Yale still at the time, doing a comparative single-cell transcriptomic study with a range of mammals, going all the way to the marsupials, primitive eutherians, guinea pig and mouse, and published data from macaques and humans. That paper was published recently this year. So I think nobody needs an introduction to guinea pig, mouse, or any of the primates, but there are two more exotic species I would like to introduce a little bit. At the one hand, we have Monodelphis domestica, is an opossum species from South America, and belongs to the marsupials, so the ones that are not don't have a extended gestation. And their pregnancy is quite special, quite different from what we know from eutheria. So here you see a timeline where copulation happens on day 0, and the whole pregnancy takes about 14.5 days, so only 2 weeks. But of this day time, most of the time, the fetus is actually, the embryo is separated from the mother by an eggshell, an egg covering, and only on day 12.5 this egg covering is lost, and the time the fetus is in direct contact with the mother is only for 2 days, so it's about 14% of the gestational time. So you can see this here in histological evidence. So here is an 11.5. This is the epithelium of the mother. This is the trophoblast. And here you see this extracellular layer that we call eggshell or shell cover. And on day 12.5, this layer is gone, and the trophoblast cells are in direct contact with the uterine epithelium, and then all kinds of stuff is happening here that I don't want to talk about. So if you compare this, for instance, with a mouse, much smaller animal, implantation happens at day 4, and the actual placenta exists for whatever 17 days. So 80% of the gestational time is in direct contact between the mother and the fetus. And in the case of humans, it's actually 97% of the gestational time with direct contact. So we have here in this phylogeny here, placentation coming probably arose somewhere before the common ancestor of marsupials and Afrotherians. By the way, this is a marsupial, but it doesn't have a marsupium. Therefore, it's called Monodelphis, having only one opening, namely the cloacal opening, instead of 2, which would be the pouch. So the youngs are actually hanging off the belly of the mother. So, so much about the opossum, which is the only really well-developed laboratory marsupial that we have at our hand. The other one is Tendra ecaudatus, which is comes from the common ancestor of eutherians. And what's happening between here and here is that here sustained placentation evolved. Here we have placentation of 2 days or up to 8 days, while here gestation can last up to 2 years. And okay, and that has consequences, the consequences that I mentioned before. In general, marsupials are relatively undeveloped. In eutherians, they can keep the fetus inside long enough so that they can more or less fully develop. So this is the case. That means that the interesting evolutionary innovations actually happened between this node and this node, which is the stem lineage of the eutherians, because what is interesting is the ability to have the sustained fetal-maternal relationship. And in order to know what's going on there, it's necessary to include in our comparison animals from the most basally branching part of the Eutherian tree, and those are Afrotherians, so tenrecs, elephants, and hyraxes, and things like that. And in fact, this is the reason why we chose to work on tenrec, in particular tenrec ecaudatus. So they are Afrotheria, as I already mentioned, so they are the most basally, most primitive, quote unquote, Eutherians. They have a bicornate uterus. This is not a tenrec uterus, it's a mouse uterus, but morphologically looks the same. Interestingly, they have a cloaca, which you usually would expect from a marsupial, a bird, or a lizard, or any reptile. It's quite surprising to have Eutherians, proper Eutherians with a cloaca, but nevertheless, they have the most advanced form of fetal-maternal relationship, namely a hemochorial placentation, where the fetal cells are in direct contact with the maternal blood. And this was possible through a collaboration with Frank van Breukelen at the University of Nevada, who has a breeding colony of tenrec ecaudatus animals. Until our work was done, actually, Daniel Stadtmauer initiated that. The only trace of the placenta of tenrec ecaudatus was one paper from 1936 that was based on a specimen of a single animal that was dug up by a traveler in Madagascar and killed and fixed, and then went to Switzerland, and then Götz actually got this animal to work on. And the only thing we knew were this histological picture that they have identified as potential residual cells, only based on morphology. So this is how this animal looks like. This is the female reproductive tract of a pregnant animal. With each of these little grapes here are implantation sites and embryos. This is the vagina, is the urinary bladder. And oops. So, yeah. Here you see a cross section. This is the embryo, of course. Here you see the neural tube and so on. And this is the placenta and the decidua. And this is the myometrium around the thing. So this allows us to have some glimpse of what happened between the theory and the eutherian placentation by including this animal together with the more usual model species, lab species like mice and so on, which unfortunately actually don't get us very deep into the phylogeny of mammals. Yeah, okay. Okay, so now it's also important to remember that these key innovations that were happened in the stem lineage of Eutherians had to overcome quite a number of cell biological obstacles. For instance, Hans-Jörg Denker pointed out that attachment of the uterine epithelium with the embryo is unexpected because epithelia are not supposed to stick to each other. We pointed out that there's a so-called information paradox, which are talking about. Best known is Paul Medawars' realization that there's an immunological paradox because the embryo should be attacked by the adaptive immune system because it's genetically different from the mother. And there's a more recent discovery also, namely the fact that the placental bed is actually an open wound. And one of the key innovations is to prevent the healing of the wound. Otherwise, the placenta would detach, and that would be a problem. Okay, the inflammation paradox simply comes from the observation that the embryo, when it makes a placenta, is actually invading the maternal tissue, causing an injury to the maternal uterine lining, and any disruption of an epithelium could generically cause inflammation. I think anyone who has done anything in medical sciences knows that, and that would be a problem because the neutrophils would be activated and they would attack the embryo, and that is independent of the immunological status of the embryo. That is just a generic reaction to the injury that's caused by the embryo. Just a little bit of terminology. I already talked about hemochorial placentation before, but there are other forms where, for instance, the embryo is directly contacting the epithelium of the uterine lining of the mother. That's called epitheliochorial, which we find, for instance, in pigs or in other farm animals. And then an intermediate form where the luminal epithelium gets eroded, but the blood vessels of the mother remain intact. It's called endotheliochorial. But as it turns out, the most invasive form of placentation is actually the one that evolved before the common ancestor of all eutherians. So the hemochorial, the most invasive form, is actually primitive for eutherian mammals. Okay. Well. So then the question is, so how was it possible to overcome these obstacles for the establishment of the fetal-maternal interface, in particular wound healing problems and inflammation, and in part also the immunological paradox? Well, we believe that the key innovation was actually the evolution of a new cell type, or actually a cell type family, as it will turn out. And this is the decidual cell, which is here. You have a decidual stroma cell in a human, endometrium here. And this cell type evolved in the stem lineage of placentas. They don't exist in marsupials. Okay, so now I'm coming to the actual. That was the preliminaries. For those who are not thinking about fetal-maternal interfaces all the time, I want to talk about two areas. The one is origin of novel cell types from the embryo, the trophoblast cells, and the decidual cells on the maternal side. And then I want to talk about communication network evolution between fetus and the mother. And 2 points. The one is prediction about disambiguation. And then I will talk about that there's very little evidence for fetal-maternal conflict in the evolution of the fetal-maternal relationship, which is actually unexpected, at least from some points of view. Okay, so first let's talk about trophoblast cells. Trophoblast cells are the blastocyst or the embryo and the fetus, the outer layer of the embryo where it contacts the mother, and it continues to be the cover over the placenta, the cells that are in direct contact with the mother. Oh, come on. Okay. Maybe it needs a better battery later, the next talk. Okay, so this is sort of the overview of gene expression differences and types of trophoblast cells that we found in our comparisons. Let's start with monodelphis. There we find so the yolk sac. We don't talk about yolk sac, but two types of trophoblast cells. The cytotrophoblast CTPs, as well as what we call as trophoblast giant cells, which are actually quite interesting. Also later on, this is now going back. And these cells you see here, one of these giant cells, and what they are doing is actually quite interesting because they are producing a lot of inflammatory cytokines like interleukin-1, interleukin-6, 17, prostaglandins, and also VEGF-A, signaling to a bunch of maternal cells. And we'll come back to this later again. Okay, so this is what we see in opossum. In the others, we find another a new cell type coming in, at least in terms of its behavior, that we call EPT, so extraplacental trophoblast. Those are cells that go out of the maternal tissue and invade the maternal tissue. And we find the expression will come in a second. I mean, they are called extraplacental because we know they are outside the placenta, but they are distinguished in terms of gene expression, as you can see here, from other trophoblast cells. So the fact that we talk about this cell cluster is based on transcriptomics, but extraplacental nature of them is anatomical. And you see them also in primates, also in humans. This is too complicated for me. At least I cannot talk about it here in a comprehensive way. But the main thing is that extra placental trophoblasts arise with eutherians. Come on. Just wanted to say, sort of in the human, this extra placental trophoblasts arise from the cytotrophoblast and the anchoring villi and invades the endometrium, and also give rise to this bile artery remodeling that ensures that the maternal blood vessels are open and provide the maximum amount of blood and nutrients to the mother. Then we have another sort of morphological type coming up, the syncytial trophoblast that we find in human and the macaques, but also in the rodents, but not in the tenrec. You'll see this one is actually also a syncytial. So syncytial, of course, means that they consist of fused cells and making the cover of the placenta in humans, also in mice. And the interesting thing here is that this ability to fuse comes from a gene that is captured by mammalian cells or other vertebrate cells from retroviruses. Syncytin, as these proteins are called, are originally envelope genes for retroviruses that they use in order to get into the host cell. If this gene gets integrated into the host genome, they can also express this envelope gene, which is then called syncytin, and this can then be used to initiate cell fusion. So the evolution of the syncytial forms are derived from virus derived gene. Okay. Yeah. So and one surprise that we had is that these EVTs, extraplacental or extravillous trophoblast, which is a cluster of similar cells from different species, is actually clustering with the monodelphis triant cells that I already introduced to you. There's another one. This is an invasive syncytial cell in the guinea pig. So this extraplacental trophoblast is homologous to the opposite trophoblast triant cells, which themselves are not invasive. It's actually interesting that this invasive cell is syncytial. And if you look at the distribution of trophoblast cells with fusion, it turns out that we find them here, everywhere, except that if you look at the homology groups, syncytial forms actually come in different subclusters, which suggests that syncytialization actually evolved multiple times independent. So one argument here is, for instance, the primate syncytial trophoblast 1, as we have called it, is actually seems to be homologous to the cytotrophoblast of the mouse, or the syncytial trophoblast cell, and the mouse seems to be homologous to the cytotrophoblast in primates. So that suggests that syncytialization evolves multiple times, and this is going the wrong direction. Even though and it actually turns out that this capture of retroviruses with this mantle protein happened multiple times independently, so that in the primates, two events where transitions were evolved or invaded the primate genome independently. We see the same event in rodents and in rabbits and lagomorphs and so on. So our conclusion that some of the syncytial cell types that we identify are actually not homologous to each other is consistent with the independent recruitment of these syncytious genes in different lineages of eutherians. Okay. So now let's move on to the decidual cells, which are actually the most important cells. This, I made the argument that they are responsible for evolving a maternal tissue that is able to tolerate and accommodate the placenta from the fetus. So here is what we see similarly in comparison in the opossum. We've identified two fibroblast-like populations. TF is tissue fibroblast, so that's probably just a normal fibroblast that makes collagen and from the but they do have an endometrial fibroblast that is different from the tissue fibroblast, and that is also homologous to the others. So what is then, this is consistent with what we have shown earlier with immunological methods. There are these cells under the epithelium that are probably homologous to the endometrial stromal fibroblast of eutherians. Now let's look at the comparison between opossum and tenrec. Tenrec, we see three populations, one of which I'm not sure it's actually real. But it's, no. Okay. This is only a minority. We have again an ESF population of endometrial stromal fibroblasts that are similar to those. And then we have the first form of the decidual cell, which we call IL-15, because they have this characteristic there. We can see from what's called RNA velocity analysis that the endometrial stromal fibroblasts from tenrec have a ontogenetic trajectory to become these decidual cells, this IL-15 expressing form. So they are, if you want, a thermal addition to the fibroblast differentiation cascade, another step towards a new cell type. That is the tenrec decidual cell. So there's also, this transition is also coincidental with a major change in gene expression profile, where a lot of the typical genes that you would expect in the decidual cells get expressed, but not before. So we interpret this that the monodelphis and the marsupials and the Therian ancestors have what we call a paleo-ESF, namely the one that cannot differentiate into a decidual cell, and then in the stem lineage of eutherians, it transformed into what we call a neo-ESF, which has the ability to differentiate into a decidual cell that helps maintain the placenta. Then the question is what's happening here as we evolve further to more derived mammals. And there, so here is the tenrec with these two forms, and now we have actually a number of additional decidual cells identified here in purple that are all characterized to be more endocrine, produce prolactin, for instance, or oxytocin and things like that. So maybe so there is this also in the human, there is an indication that there are sort of these three populations of the sexual cells, or in the mitral chromo cells. There is the one, this is data from a pregnant woman, that are these activated fibroblasts because they express smooth methylactin. Then the second population already expresses a typical vesicular gene like interferon gamma, insulin-like growth factor binding protein 1. Interferon gamma has to do with my work on pig endometrium and just working on, sorry for that. And that is shared between this population, this, and then the cells down here are what we call mature testicular cells because they express, for instance, prolactin. So these would be the in humans the endocrine cells, the pre-residual cells, and these would be activated fibroblast cells. Maybe I shouldn't go into too much detail, but the gene expression correlations also show that these pre-decidualized cells and the mature cells are actually in the gene expression profiles anti-correlated, which is the indication that they are actually real cell types, different cell types. So then the answer is that we have paleo-ESF, neo-ESF with the pre-decidualized cells, and then endocrine decidualized cells evolve after this. This is sort of a nicer way of putting all of this together, where we have sort of three layers of cell types. We have the endometrial stromal fibroblasts. Then we have these pre-decidual cells that we only not find in mid-gestation in the mouse, but in all the others. And then we have another bunch of homologous cells that are all sort of this more endocrine variety. And actually, not the human endometrium is the most complex one. It is actually the rodent one that even has more cell types, at least cell populations, than the primate. Okay, now let's move on to the cell communication evolution, which is of course a view that we can only now with single cell data approach. And the basic idea about reconstructing communication networks comes from this paper here. Michaela has realized that if we know which cell produces a ligand and which cell produces a receptor, you can infer a potential communication event between these two cell types. So this was actually the first reconstruction from human placenta, term placenta. This approach was then industrialized by the Sanger Institute in the cell phone BP programming software package. Okay, the 2 points I want to make with using this inferred cell-cell communication networks. One is, I will argue there is a trend for disambiguation. You know the term disambiguation from using Wikipedia. If you put in the name, for instance, Schröterer, there might be many people having this name, and then it gets disambiguated who is whom you are actually looking for. So in this case, a signal or a ligand means different things biologically when it's coming from the mother versus the fetus, right? The fetus usually wants to have resources, the mother wants to regulate what's going on. So the same molecule can mean different things whether it comes from the mother or the fetus. And so a signal from the fetus is, whatever. And the idea is that that after mother and fetus start communicating with each other, there should be an evolutionary tendency of producing ligands preferentially either in the fetus or in the mother. And this is exactly what we find. Daniel Stadtmauer, who was one of the leading graduate students in this whole study, together with Silvia Basanta, he developed a test that essentially looks at the distribution of fetal only, maternal only, or both ligands, and then does a chi-square test in order to see whether it's deviating from the null expectation. Of course, the null expectation is that most of the ligands should be on both sides for obvious statistical reasons. And what he found is actually pretty strong evidence for disambiguation. So here it's sort of expect, so this is co-expressed ligands in a particular family of ligands, like Wnt or steroids or somatotropins and so on. And the number of the expected number and observed number, when if they would be the same, would be this line here. You see you have systematically fewer ligands that are expressed by mother and the fetus than expected by a statistical comparison, or the same observation the other way around. Fetal-only ligands are more frequent than expected by chance, and maternal-only ligands as well, overwhelmingly within one group of a family of ligands. You can also put the whole thing on a phylogeny and ask what is the evolutionary trend of getting ligands being expressed in one or the other tissue, in order to make this a little bit easier to see. So this is here the transition matrix from so F0 M0 would be not expressed at all. This is only expressed in the mother, it's only expressed in the fetus, and this is expressed in all of them. And this gives you the ancestral state and the derived state based on our reconstruction. And you can see, okay, yeah, that the majority of the transitions from 0 to not expressed at all are to disambiguated states. So 70% are either in the mother or the fetus, but not of both, and only less than 30% of the cases it evolves to a ambiguous situation. In the same it's the wrong direction. Okay. And if you look at evolutionary transitions where a ligand was expressed both in the fetus and the mother, in the most cases it evolves from a co-expressed one to a disambiguous state where over sort of 68% of the transitions are towards not in the fetus but in the mother, and the other way around. So that suggests that there is actually a trend of sorting out signals as being either fetal or maternal. Okay, the next one is probably the one that you are most interested in. I will make the argument that there is a little evidence for fetal-maternal conflict. Now, fetal-maternal conflict is the idea that evolutionary changes in the fetus can benefiting the fetus can disadvantage the mother and the other way around, so that there is sort of a tug of war over resources and control of parturition and so on, where the evolutionary interests, Darwinian interests of the mother and the fetus are in disarray, and actually would expect to go into try to pull in different directions. Now we looked at our data and made the following idea. So if there would be conflict, you would expect, for instance, that if in evolutionary time the fetal ligand is increasing in expression, the maternal receptor go down because it's not in the interest of the mother, right? It only benefits the fetus. Therefore, the mother has an incentive to decrease the efficiency of that signal by decreasing the receptor expression on her part. And in the comparison, so where you have no evolutionary time, you would expect a negative correlation between fetal ligand expression and maternal receptor expression. In the case of cooperation, meaning that changes in ligand expression is in the interest both of the mother and the fetus, one would actually expect that there is either no correlation or a positive correlation between fetal ligand expression level and maternal receptor. And in the comparison, we would expect a positive correlation between expression level of the fetal ligand and the maternal receptor. And now, of course, now the upspringt ins Wasser. Here are the results. If we put, if we look at our all our data and the evolutionary changes over the time of Eutherian development, we find that by far the majority of signaling connections actually evolve in the way you would expect for cooperation. There are a handful of cases where there's a negative correlation, and these are actually cases with, for instance, this IGF-2 and IGF receptor that are negative correlation, where you, based on other evidence, we already know that there is evidence for conflict in this area. So, of course, conflict signals exist, but it is actually a relatively rare occasion compared to the evolutionary changes that happen with other ligands, where the evolutionary changes seem to go in the same direction, being consistent with the idea that fetal and maternal interests are aligned or cooperative. So this is what I just said. We can now do the same thing for maternal to fetal signal, and the pattern is exactly the same. And this sounds like it contradicts the conflict theory. And I will tell you that this is not the case, because if you look at conflict mathematical models actually closely, and that was also pointed out by David Haig himself, the Pope of fetal-maternal conflict theory, in a paper from 2010 in a book. And here this diagram is actually quite interesting. So F means an mutational change that benefits the fetus. It goes in this direction. This benefits in this direction. Mother going in this direction. So any mutational change that goes in that direction is benefiting the mother. And this are the area where would be mutations where fetal and maternal interests are aligned. And that is even the case when, let's say, we have a mutation that is good for the fetus but bad for the mother initially, because the mother has still 50% of her chromosomes paired with the fetus. So they can benefit from, even if he as an individual has a disadvantage, genetically can be advantaged, because this change to the fetal biology can also lead to the selective advantage of the maternal genes that are also part of the genome in the fetus. And David makes the point that actually the real conflict only happens in this little pizza slice in the overall space of possible mutational effects, which qualitatively says most of the time mutational changes should benefit the fetus and the mother, and only a minority of mutations that fall into this particular two pizza slices here should show the signature of conflict, and this is exactly what we see here, right? So I found this actually a quite interesting observation. Now with this I come to my conclusion, not too long. So the fetal-maternal interface is a focal point for the evolutionary origin of new cell types. That is true both for the fetal cells in the case of the trophoblasts, like syncytial trophoblasts, extraplacental trophoblasts, and so on. And on the maternal side, monomodulatory cells like the pre-decidual cells, endocrine decidual cells of different forms, in particular in the rodents. And in terms of the fetal-maternal communication, there's a tendency for disambiguation, correctly predicted by David Haig, and also predicted by David Haig, but not realized by a lot of people who follow his lead, is that the fetal-maternal communication is mostly driven by cooperative co-evolution rather than conflict. And with that, I say thank you to all the people who did the hard work, and I thank you for your attention.